So good evening. We are live. Happy belated Thanksgiving to everybody. We're live here at Icaria Concierge and Functional Medicine. We're very excited tonight. Dr. Brett Haake. I'm Dr. Brett Haake. I'm here with my wife, Dr. Felice Haake.
Hello.
And we have a special guest with us tonight, Dr. Val Zudans. Dr. Zudans is an ophthalmologist here in town. And before we kind of get to the main topic of what we want to talk about, let me just kind of introduce Dr. Zudans. He is currently the owner and the owner-operator of Zudans Eye. They're out of Vero Beach, Sebastian, and also Okeechobee, okay? His education, he did his fellowship at Baskin Palmer Eye Institute. He did his residency at Thomas Jefferson University, and he's a graduate of University of Florida College of Medicine and also the University of Florida undergrad. And he also, very cool little shout out. He is the founder of the biotech company InSurf LLC, and here's his Methylene Blue product right here, which we carry at Icaria, and there's a lot of other locations that also carry this product. It's a fantastic product. So what we're going to talk about today, if you've been a patient at Icaria or you've seen our podcasts in the past, or just search us online, we emphasize and we focus a lot on metabolic health and insulin resistance, and by extension, you've probably heard us talking about mitochondrial health. And so it's interesting today that we've got myself, conventionally I'm an anesthesiologist, my wife is a family practice doctor and medical bariatrician, and Dr. Zudans, as we just stated, is an ophthalmologist, and we're going to actually talk about metabolic health. So it's kind of curious that we're talking about that. And so what I'd like to start off with, first, Val, welcome to the show.
Thank you, man.
Thank you for coming on.
Good to be here.
What I'd like to start off with
Been looking forward to it all week.
Oh yeah. If you know Val and I, our wives laugh because we've nerded out on these types of topics literally for probably the last couple of years. I can remember I was working up in Vieira. It takes me about an hour to get up there. And I think we talked about the mitochondria for the entire ride. I was on 95 for an entire hour. We literally just talked about the mitochondria.
You came by my house to drop something off. It was supposed to take like a second. And we were out on my front porch talking for
about an hour. I think we ended up solving a lot of different philosophical conundrums.
Yeah, I've seen their text message strings. Yeah, they're like novels.
Yeah, right. The Aristotle, Plato painting. We were getting to the bottom of all that. We solve a lot of issues. So I think it's interesting. Tell me first, how did you as an ophthalmologist get involved with wanting to know about metabolic health and the mitochondria?
I didn't think I had anything to do with that stuff. I was like 10 years old, and I was looking through some magazine about day in the life in the Soviet Union, and they were talking about this guy Fyodorov who had these assembly lines for doing RK surgery back in the day to fix people's vision. And I thought that was the coolest thing in the world. So that you could cut someone's eye and actually make them see better. So I always thought I was a surgeon, and I'm more like a craftsman, carpenter than a physician. And that's actually how way back when it used to be, like Hippocrates' time, it was you had physicians and surgeons were not the same thing. And anyway, so I went to medical school, kind of a little bit of an engineering background, and I switched into chemistry because I wanted to get into medical school faster. I didn't feel like wasting my time doing engineering. Yeah. And I went into medical school. I didn't know what diabetes was. Okay, I knew it had something to do with sugar, but I didn't know anything. Right. And it actually turned out great because I'm very curious, and so I'm going to class, and everyone else thinks they already know what diabetes is and all this stuff, and I'm just listening and I'm learning stuff. And then I went into practice doing cataract surgery, doing all my surgical stuff, still thinking I'm a surgeon. And then actually I read a book Kate Shanahan wrote. She was a family practice doctor out in California, Deep Nutrition. And I started reading through this. I'm like, holy, I started understanding that you get like 3 hours of nutrition in medical school, and it's the most important thing to your health. Okay. And and then I kind of got, at the same time, I started just getting curious in like the biohacking people, Peter Attia, and reading and watching them. And I started, trying all this stuff on myself, trying to make myself healthier. And so somehow along the way, I started to figure out that I'm down this, all medicine is sort of super subspecialized into these areas. And we think we're just like one thing, but it turns out we're all treating the same disease. I don't care what part of the body you're dealing with, you're treating metabolic dysfunction. And it may not be that you're treating the active metabolic dysfunction right at that time, but you're treating, in the end stage results of all that. So something simple like cataract, okay, we think cataract is, your lens got cloudy. It's just because you went around the sun 72 times, and therefore your lens turned cloudy. And then I go in, I'm my carpenter, I clean out the cloudy lens, put in a new lens. That's cataract surgery. But that cataract is very basically, it's oxidative damage over time. And that is all metabolism. And it was kind of curious because I'd have patients, who were 80 years old and they barely had a cataract. And I'd have 55-year-old diabetics who had the worst cataract I've ever seen. Yeah. They're not aging at the same rate. So that I started to get curious. I'm curious. And so then curiosity killed the cat. I ended up like just diving deep down into these things of trying to understand, how does biology work? Not really even, we're humans, but the same thing is going on in all biology. This oxidative damage from insufficient energy production in the cell, in our cells, in mitochondria.
Yeah. And you bring up a really interesting point. I know with Felice's practice, with our practice, one of the things that we are constantly stressing to our patients is that we talk about insulin resistance. And so we say, okay, resistance to insulin is just simply stated, it's the cell's inappropriate response to insulin signaling. But we say the reason we want to measure insulin resistance is that it's upstream from all of these non-communicable diseases in the body. So the ones that are kind of low-hanging fruit, obviously type 2 diabetes. Now we're starting to understand Alzheimer's is, we call it type 3 diabetes, but it's really advanced insulin resistance of the brain. You got fatty liver, you've got coronary artery disease. Over 50% of everybody that gets diagnosed with type 2 diabetes already has plaque in their coronaries, peripheral vascular disease, endothelial cell dysfunction, sexual dysfunction. The list goes on and on and on. There's actually a psychiatrist out of Mass General who's talking about, basically most psychiatric diseases are essentially metabolic diseases. Yeah. What I find to be interesting with what we're talking about today is how all of these chronic eye conditions are actually related to metabolic disease. And when we say insulin resistance is directly related to mitochondrial dysfunction. They're highly, highly related. Specifically, when we say resistance to insulin, there's a problem at some point along the chain of where, when insulin hits a receptor called insulin receptor substrate 1, there's a whole series of reactions that occur that ultimately results in something called the GLUT4 transporter moving to the surface of the cell, and then glucose moving into the cell. When that process is efficient, we call that insulin sensitivity. When there's a problem with the process, we call that insulin resistance, but it's directly related to mitochondrial dysfunction. And so what I find interesting is how all these different chronic eye disorders are actually related to these different things. So one of the first things that I wanted to talk about.
Reaffirm that, we're all just treating the same disease. The same disease is your cells, you're getting too much radical oxygen species damage. It's oxidizing the enzymes in your mitochondrial electron transport chain. The chain is not working. You're not producing enough energy. And then all aging is oxidative damage to your mitochondria. Yep. And so I don't know as much about the pathways as you do. I just think of insulin resistance as it's just a tolerance. It's just like any other drug tolerance. If you keep bombarding insulin receptors with too much insulin, it's hyperinsulinemia over time leads to tolerance. The insulin hormone doesn't work as well. That's what insulin resistance is.
Exactly. Right.
So, and it's very simple. It all goes back to your food that you eat.
Right.
If you don't eat foods that cause your pancreas to release insulin, if you don't get spikes in insulin, you're not going to get exposed to that much insulin. So it's insulin under the curve. The volume of insulin you get exposed to during the day is what causes insulin resistance.
Correct.
That's why it's the exact opposite of what everyone, conventional wisdom, thinks is correct. They're telling people snack all day, don't ever be hungry. It doesn't matter what the hormonal response to the food is. They're basically saying carbs are the same as protein, is the same as fat. No, they're not. They have different hormonal insulin responses. Right. And so you have to and it's like even things like methylene blue, it's great. But if you don't fix your diet, that's like more than 90% of your health. When you eat, what you eat, how you sleep, circadian alignment. That's it. Yeah. That's like 95+%. And then once you're doing that stuff, then you can start tweaking things like,
it's as if you're echoing what we say. We talk to patients and we say, okay, there's all these cool things that we do in the functional medicine space. A little shout out to InSurf. Methylene blue is one of the things that Val's talking about. But there's all these really cool things: photobiomodulation therapy, the various types of peptides, optimizing hormones. There are all these really, really fun things that we do that will make you feel better, that are going to give you energy, that are going to, you potentially lose body fat, gain muscle, all these kinds of things. But if that foundation is not there, just like the chair that you're sitting on, if that foundation isn't there, the chair becomes worthless. And that foundation is diet, exercise, sleep, stress. And to your point, diet is a huge part of it. And then, movement. We make all this stuff about, okay, resistance training or CrossFit or this or that or whatever, but it turns out and this is what we talk about all the time with our patients, is that we might have a 60-year-old patient who spent his or her life lifting weights and surfing and doing marathons and all those kinds of things. Now that person may want to be a little bit more active. We've got other folks that really have never worked out. And what we try to do is work with the patient, say, hey, listen, you've got to move. You've got to move. You've got to learn how to move. And you've got the guy in Sardinia, okay, or Okinawa, or all these blue zones, Icaria, right? They're not looking, the 100-year-old, the centenarian's not looking back to the United States saying, oh, let me try this most latest and greatest CrossFit workout, and then I'm going to be 100 years old. They're doing things that are cultural, that they've done for centuries, and that's things like walking up and down hills. They're not on social media with all the toxicity and all those kinds of things. They're eating grass-fed lamb that's farm to table with the vegetables. They're close with their family. They're going to church on Sunday, and it's shocking. All these things lead to longevity, and so it's really interesting. But I want to get down into the nitty-gritty.
Okay.
Let's talk about, not to put you on the spot, what I would like to talk about.
Is there going to be a quiz here? I don't know if I'm going to pass.
I got to tell you, if you're in medicine, if, for instance, like I said, I'm an anesthesiologist who works as a functional medicine doc. My wife's a family practice. If you're in medicine, the last time you looked at a lot of these different eye conditions in a nuanced way might have been when you were in medical school. If at all. So
I don't remember much at all in medical school about ophthalmology.
No, it's one of those. It's a very.
I have the coolest from metabolism. I have the coolest field in all of medicine because my organ is the most metabolically active, the highest mitochondrial biodensity. I have the coolest toys too. I can measure electrical potentials in cells just by this test called electroretinogram. It's really cool.
So talk about that a little bit, because you had mentioned that before. Yeah.
Very simple test. It's like a EKG, and the printout almost the waveform looks just like an EKG. So It's just a flash of light in front of your eye. There's a little sticker sensor, like an EKG sensor, below your eye, and there's three points to the wave. Okay, and you get latency and amplitude. So latency is the amount of time from the flash until the signal starts. Okay, you want that to be fast.
Okay,
and then there's an amplitude. How strong is the signal? And
you want the amplitude to be.
You want to be as high as possible.
Yeah. So,
and it gives you like
short latency, high amplitude.
Right. Okay. And I get percentile printouts.
Okay.
Age adjusted percentile printouts for
interesting.
3 signals. First signal is light hits your photoreceptor. The photoreceptor is polarized and it gets depolarized, and it gives the photoreceptor signal, and they're separated out on the. They're able to look at the 3 of them. Second signal is bipolar cells. So in your retina, the back layer is the photoreceptors. It's a transparent tissue, so the light goes through to the photoreceptor, and then it sends the signal to the next cell, which is the bipolar cells. Okay, and they're doing it's like a computer circuit. It's like processing it, but you get a signal from that. And then the third one is ganglion cells. Ganglion cells are the cell nucleus for the neuron that runs from the retina back through the optic nerve to the lateral geniculate nucleus, the midpoint of the brain. So there's only 2 neurons between the retina and the back of the brain.
Wow.
And there's also another test that I don't routinely do, but called a VP. The neurologists do a lot of it, visual evoked potentials, where they can measure that third transmission from, you put some probes on the back of the head, and you can measure how long it goes there. Yeah. You get slowing and things like multiple sclerosis, where the optic nerve is inflamed, and so it doesn't send the signal as fast.
Yeah.
So it's really cool because latency is slow signals mean current stress on your cells, and low amplitude means damage to the cell.
Wow.
Okay, so I can see, sometimes I'll see a glaucoma patient. Glaucoma is a disease where I think it's more complex than the way we think of it, but we think of it as high pressure causes damage to the nerve. And it's actually ganglion cells that die off. You actually lose volume of ganglion cells over time with prolonged damage. But the high pressure creates stress, so you can slow the signal. And in the early stages, you can have a delayed signal, normal amplitude still, because they haven't had damage yet. But the cell's under stress.
So that'll tell you that it's still salvageable if you get the pressure under control, right?
Not only is it salvageable, I've actually measured this before, where I'll do an intervention, like do a SLT laser or glaucoma drops, lower the eye pressure, take the stress off of the eye, the signal speeds up.
Wow.
So latency decreases.
Latency decreases, and then you know that you've taken away their risk. Because glaucoma is a very slow disease. It takes a very long time. For a lot of all these chronic diseases, they've been festering. It's like 50 years or 60 years of abusing your body, and then you have cancer.
Right.
Kids normally don't get cancer. I mean, they can, but it's more likely that you have breakdown of systems over time, and then eventually something emerges.
Yeah. Right.
And even like all the chronic diseases all in our body, like psychiatry, there's a great book on psychiatry that came out called Brain Energy that goes into all relatively recent. Harvard professor.
Yes,
his
that's
name
the same
is
Chris,
guy. Chris Palmer? Or Chris,
he spoke right after Thomas Seyfried back in the fall. There's a great
he's
podcast.
Awesome. Great book. But in psychiatry, different people will end up having these aged phenotypes later on from chronic metabolic dysfunction. And the reason why I might get, if I have bad neuropsychiatric disease, I might get schizophrenia, or you might get Alzheimer's,
depression,
or so you might get Parkinson's. It's because we have genetic diversity, and the weak point in our systems, it shows up as this age-related disease. They're all the same root cause, but it's just showing up as different phenotypes. So
this is fascinating, because that same psychiatrist spoke after a guy by the name of Thomas Seyfried, and you and I might have talked about him before. but Thomas Seyfried is a tenured biochemist and geneticist out of Boston College, and his entire work is he's taken the work of Warburg, who was the German scientist who figured out that cancer was a glucose fermenter. So without any knowledge of the mitochondria, and really without any knowledge of DNA or the electron, no advantage of the electron microscope, in the late 1930s figured out that cancer essentially makes ATP by converting glucose into lactate. He had no knowledge of the mitochondria.
And can only do that.
And only do that. So what Seyfried has demonstrated over and over again, what he's demonstrated over and over again is he takes all these different kinds of cancers and he shows that the mitochondria in the cancer cell is completely dysfunctional. And he takes the mitochondria from the cancer cell, puts them into normal cells, and the normal cell then develops cancer. Conversely, he takes the DNA from a cancer cell, puts it into a normal cell with normal functioning mitochondria, and that normal cell stays normal. And so his entire thesis is that cancer is essentially a metabolic disease. It's a disease of the mitochondria, and it's not a disease of the DNA. And to your point, we were talking off air a little bit, the mitochondria, when it becomes dysfunctional, when it becomes oxidized, and you have all these reactive oxidative species, electron leak, that then affects the DNA. And then we're looking at the DNA and we're like, oh, well, this cancer here, this disease, it's a genetic disease, when in reality it was actually a metabolic disease, and we need to focus on the metabolic aspect of things, I think, more so than the actual genetic. And this gets into this whole idea that
it's not a genetic disease. So every day of our life, we probably have one or two oncogene mutate, like that would lead to death. Okay, every day of our life, but we're not dying every day of our life. And I think it's primarily a breakdown in immune function, which is also age related. So what happens is when you start to form a cancer, your immune system, if it's healthy, it actually encapsulates it, or it kills it off. And so when you have chronic poor metabolism, you just let down your guard. Your killer T cell, all that stuff is not working.
1,000,000%. So tell me again.
You know, Richard Nixon declared war on cancer, and right after they did the Human Genome Project, they did this the Cancer Genome Project, and they basically found every single oncogene in your body, thinking that all you have to do is figure out the countermeasure against these gene defects. And they didn't cure, they cured one, I think it was chronic myelogenous leukemia, basically had a cure. They thought they were going to be able to find every single other one. They haven't had another single one from genetic. They figured out almost every single major gene defect, and none of them led to curing cancer. Yeah. And it's kind of laughable. I saw this press conference. They had Larry Ellison from Oracle, and they had the guy who's the head of SoftBank, the Japanese, does all this investing and all the AI and stuff like that. And they basically said they were going to spend $500 billion and they were going to cure cancer using AI. Okay. I can guarantee you that, I don't think that they're going to cure a single thing.
Problem is because all the input is focused. It's human input.
They're thinking that it's a genetic problem, and it's not a genetic problem.
And that's exactly, Seyfried's funny when he talks. He's like, everybody's talking about curing cancer and standing up to cancer, and we want to throw money at cancer. And then he goes, and you actually find, he's actually got a couple. Now, he's not saying he's got the cure, but one of the things that he does is he thinks every cancer patient should be on a neoadjuvant keto diet. And what he does is he's got a couple of glioblastoma multiformes that have lived outside of 5 years by putting patients on the keto diet, which is, if you know about GBMs, that's phenomenal. All kinds of other disease processes. My grandfather died from it. Yeah, all kinds of other disease processes. And the whole thinking is that ketones enter into the cell via an entirely different mechanism than than glucose. Insulin hitting insulin receptor substrate 1. And essentially, and the other thing is that ketones are highly, highly efficient at oxidative phosphorylation, and they give your cells that metabolic flexibility. And when I say oxidative phosphorylation to the audience, I mean what we're talking about, the electron transport chain. And just to clarify, the electron transport chain is a complex of 5 different proteins, and it's essentially an assembly line as far as how your mitochondria, and specifically the mitochondrial matrix, makes ATP. And it does it by transporting electrons across, from one protein to the other, 5 total. And these electrons, what they do is push protons into an area called the mitochondrial membrane, and there's a proton gradient that is then created, and the 5th part of that chain, called ATP synthase, uses that gradient almost like a hydroelectric dam to make ATP from ADP and phosphate. And so what is interesting is that ketones are really, really efficient at doing that. And the other thing is that when you're in a mild ketosis, you're essentially starving the cancer cells of glucose. So now the cancer cell can't make ATP, so it can't get energy, and you're giving your body ketones, which for your good functioning mitochondria are what they want. As a corollary to that, what's really fascinating is they've done studies with freedivers, professional freedivers that can all hold their breath for 6, 7, 8 minutes, and they take one group and they don't eat for 12 hours, so they're in a mild ketosis. And the other group, they feed normal meal 2 hours before they eat. They've done study after study after study, and these are all professional freedivers that are randomized to 2 groups. And the group that's in ketosis can, on average, hold their breath for a minute longer, which tells you that their ability to make ATP, their ATP production to O2 consumption ratio is higher. So all this stuff is interrelated, and so what is interesting is the take-home point here is that cancer is, if it's not exclusively a metabolic disease, it's very, very highly related to, mitochondrial dysfunction, and getting into this whole unified theory of all different disease processes like we're talking about.
Well, that is what the Warburg effect is. So the cancer cells, and there's actually a reason why the cancer cells in the cytoplasm, not in the mitochondria, they break down. They use
they convert.
They do it exclusively, and the reason is it's actually a less efficient way. It makes less ATP.
Of course, right.
But the reason why it does it, it's a rapidly growing cell, and so it's actually using those carbon units to build the cancer. And so for a cancer cell, it's good, even though they get less energy out of each carbohydrate, they can, that's the way cancers work. That's the only way that they can grow.
Interesting.
Is by doing that. And so if you don't eat carbohydrates, you starve the cancer. And then if your immune system is functional enough, it's able to destroy. It's like a battle. Like the cancer cells wants to grow. All cells want to divide. All cells want to be 2 cells. And the reason why we don't have cancer all the time is we have all these multicellular systems around that are like supervising to make our cells not divide, but they're unchecked. Yes, they're behaving more like an infection, like a bacteria that just wants to. They will
proliferate.
Forever, and it's starving the ketones long enough for your immune system to take them out.
So interesting.
Do you see that with cancers of the eye as well, like in terms of like a ketogenic diet or that same approach?
I really don't. I mean, the only cancers I see as an ophthalmologist are eyelid tumors and stuff like that. Okay. But interesting story on that. You see people with skin tags ever?
Yeah.
Okay.
Oh, yeah.
So I always thought that skin tags are nothing, right?
Insulin resistance.
Yes,
Yeah.
it's hyperinsulinemia over time. Insulin is the most powerful metabolic growth factor. It's what's causing proliferation of benign skin cells on the eye. And then I started thinking back. I'm like, yeah, all of my patients with diabetes, obesity, those are the people who those are the only ones who get it.
Yep.
And when they come in for their skin tags, I'm like, sure, I can remove them, but they're going to grow back. I knew they were going to grow back, or new ones are going to form. It's not the same ones growing back. But I never made that connection until oh my gosh. So I just had that conversation. I don't want to reveal who it was, but it was actually a family member. And
Interesting.
Well, I think of my grandpa had
not
it
my
everywhere.
immediate family, but
Everywhere. He had skin tags
Yeah.
everywhere.
Which one?
did he have metabolic
with
syndrome?
diabetes.
This is your
Puppy.
mom's on your dad's
Yeah.
side.
So I had a
Interesting.
conversation with him. I'm like, this is nothing. I'll snip it off, but you know what causes these,
Right.
Okay, and he has a good night now, but
Yeah.
Well, and it gets
it
into
was left over from years ago.
It gets into this entire idea. You and I have talked about this before. Is that really there's really no incentive economically within the conventional system in the United States to actually prevent disease, and then by extension,
No, this is
make people
monetized sick
more-
care.
Monetized sick care, and to your point, also, it's also monetized to kind of the understanding of it is the nephrologist looks over here, the heart doctor, the cardiologist looks over here, the ophthalmologist is only dealing with the eyes, the endocrinologist is dealing with the hormones, and the reality is all of this is interrelated.
Yeah.
And there is most likely a unified theory, molecular theory of all disease, and most likely it's highly, highly related to mitochondrial dysfunction. And so this is why when we say insulin resistance, it essentially means that there's intrinsic mitochondrial dysfunction. Those two, insulin resistance is more of a clinical term, but really what it is related to highly is mitochondrial dysfunction.
So I was thinking about this and related to that I think humans are much more than just our biology, but we're spiritual beings and we're a lot of other things. But from just looking at things from biology, all biology is really about is converting food into energy. And you look at, ATP synthase, okay, is the most fundamental form, like subunit of life, okay? Even bacteria, every single living thing has ATP synthase. And what it's doing is it's incorporated in some type of membrane, and there's a proton pump that's pumping protons into some space, and it's creating this energy gradient. And then it's like ATP synthase is like a turnstile.
Right.
For every 10 protons that get past, so osmosis back in science class.
Sure.
This is chemoosmosis. So water wants to go or anything wants to go wherever the concentration is lower. So there's a force that moves.
It's like a hydroelectric dam.
So those hydrogens that are in the intermembrane space of the mitochondria or in bacteria or whatever it is, they want to get on the other side of this barrier. There's a turnstile in there. And every 10 protons that go through, it produces 3 ATP. If it's in the...
Interesting.
Yes. So it's just, as it turns this enzyme is just a protein that has function. And so just by turning that turnstile, it's putting a phosphorus and it's just storing. So you have this adenosine molecule with 2 phosphates, 2 ADP. And then when you put that extra P on there, now you have an ATP, triphosphate. And then that's what's used to power every single cell. Everything, bacteria,
Correct.
plants.
Correct.
And so that's like biological life is just this, how do we get food turned into energy? And then we look at these systems that are built around it. All the complexity, and it's the most amazing, most complex
Of course.
organs. Every
Yeah.
animal, even other animals, they're just
Right.
amazingly complex, but they have all these systems. All these systems are just around how do we more efficiently convert food into energy?
Right, as efficiently as possible. Yeah.
That is your fundamental basis, and that's why all these other diseases we can trace them all back to metabolism, because metabolism. So in our mitochondria, we have this very complex, like super version of ATP synthase. ATP synthase is just complex 5 on the end. You have all these other 4 steps along the way that's basically breaking down food and converting it into energy, or pumping protons across, and then when it comes back in.
And it's so interesting that you're mentioning this, because the way that we've built our longevity center upstairs, or we're actively building, and the way that I explain it to patients when we talk about what you're talking about here with the electron transport chain, is I really talk about 3 things. And you and I have talked about this as well. As one of the things that I've always been kind of interested in as a cardiac anesthesiologist is, in the OR, it's real-time physiology. So like if I'm coming off cardiopulmonary bypass and the patient started off with a mixed venous O2 of 75%, and now I'm coming off cardiopulmonary bypass and the blood pressure is low, and now my mixed venous O2 is 50%, I know that I've got some mitochondrial dysfunction. I know I have to start delivering oxygen, whether it be to increase the cardiac output or increase the, give the patient blood or whatever, or those kinds of things. But when I explain to my patients about the mitochondria, I explain it in 3 ways. I explain when you have this electron transport chain, you create what are called, you get electron leakage because you lose integrity of the proteins, and you create what are called reactive oxidative species. The best way that I explain that to patients is it's basically almost like waste product or even sewage on the engine. So you have to clean that sewage. So we now have the ability with our Vibrant offerings to measure whether or not you've got some genetic deficiencies with, number 1, superoxide dismutase, and also number 2, glutathione, which are 2 enzymes that are highly, highly important at that membrane to clean the reactive oxidative species. Next, you're looking at the actual proteins on the actual electron transport chain and the engine parts. So do you have enough phosphatidylcholine? Do you have enough L-carnitine to actually keep that engine functional and that engine can replace its parts? And then last thing, are you fueling the actual engine? And we're fueling our engines right now by simply just breathing oxygen, but there's other things along that chain to fuel that engine. One of the things, obviously, is getting enough magnesium. Another thing is something called alpha-lipoic acid, but then another thing is something called methylene blue. So a little shout out again.
Can I explain how it works?
Yes. I'd love to. I'd love, that was what I was getting at. Yeah.
So keeping with the electron transport chain, okay, I think of it as like you're driving down the road, okay, and there's traffic jams in the road, and this is mostly, there's the 5 different complexes, but mostly in complex 1. So your car is an electron, okay, and it's going down this chain, and it gets to a bottleneck in the chain, and this happens all the time for a variety of reasons, nutrient deficiencies, not enough coenzyme CoQ10, whatever it is.
Sure. Yep.
And so the electrons start crashing off the chain. They can't get through the bottleneck. So that's what your radical oxygen species are, is there's those electrons coming off the chain. Down the road, on the other side of this bottleneck, the road, after you get through the traffic jam, it's wide open. Right. Okay, but you don't get to do that. But because it's wide open, that means those electrons aren't going down the rest of the chain. So you're not getting to the end point where you're generating the ATP. So not only are you increasing your radical oxygen species as they leak, they crash off the road, you can't get down there. Methylene blue is a very simple, it started out as an industrial dye, and it's a electron donor and electron receiver. So it's a helicopter, okay? And it's picking up electrons, it's electron receiver. Electron goes onto methylene blue, and then, but it's not nearly as tightly bound. Normally the electrons are much more tightly bound to the chain. They want to stay on the road. But so as soon as it goes onto the methylene blue, then it can drop off farther down the chain. So now you have more electrons going on the chain and you have less radical oxygen species. So it's measurable that the methylene blue in the right dose will reduce radical oxygen species, and in normal cells can increase ATP production like 30 to 70%, depending on what you're looking at.
Is it bypassing it? Is it taking the electron and bypassing where the reactive oxygen species are?
It is actually jumping off the chain,
Yeah.
going onto the methylene blue, and then getting redeposited back on the chain on a different spot.
So what he's saying is that because of its ability to do that, you get less reactive oxidative species.
Right.
Reactive oxidative species are created by the electron leak, which is created by the bottleneck. So it essentially acts as a catalyst, or a helicopter is a great analogy, grabbing these electrons and then depositing them in complex 3. Now, let's pair that with what we call photobiomodulation therapy, or more commonly known as red bed. How does red bed work, and how do you pair it with methylene blue? And then you have a really interesting case study that I wanted you to talk about as well, as far as how you treated a patient. So,
very off-label, and not recommended at home. But so the first part was
photobiomodulation.
So cytochrome C oxidase, which is
complex 4
in the mitochondrial chain. It's called cytochrome, chrome color. Red light directly stimulates the action of that enzyme, and it produces more ATP. It's almost like the photosynthesis. Basically, light hits something and it turns it into energy.
Interesting.
And so there's lots of information out there about photobiomodulation related to eye conditions, like a bunch.
I was going to bring that up. We are, as far as some of the major, eye disorders, I actually did the research here. So diabetic retinopathy, there's 42 studies on PubMed with regard to photobiomodulation therapy, 5 studies with regard to methylene blue. Macular degeneration on PubMed, 105 studies with regard to photobiomodulation therapy, 2 studies with regard to methylene blue, and then glaucoma, PubMed, 20 studies on photobiomodulation therapy, 15 studies on methylene blue. So it's. I didn't look at every study.
I gotta tell you my patient. This is pretty shocking, and I was shocked. Okay, I was in the process of doing methylene blue, and actually I didn't have my product out yet, so I found a place for my patient. She was in a motorcycle accident, had a direct trauma to her eye, and it was a few days before I saw her, and she came in, and she could not even count fingers in front of her face.
And this was a younger patient.
Yeah, this younger.
No comorbidities, younger patient.
She was young and healthy otherwise.
Yep.
So direct trauma to the eye can cause a traumatic injury, traumatic optic neuropathy. Took a picture did a 3D scan of her optic nerve, and it was like 50% enlarged in thickness, very swollen, and basically couldn't see much at all. And I wish I had done a bunch more studies. If I knew this, I would have done like ERGs. I would have done like all kinds of things on her if I knew what was going to happen. Went through the whole thing. I told her basically, the standard care, but I told her that some of this will recover because it's just due to swelling, but some of this is permanent damage because traumatic injuries to nerve tissue, you're going to have some residual effect. Mm-hmm. And she says, "Is there anything else I can do?" I said, "Well, don't tell anyone I told you this." But I actually had her, go on Amazon and buy a pretty high watt LED, near infrared LED light bulb. And I told her how to get methylene blue, and I had her take actually in the low end of the therapeutic dose.
It's like 0.5 milligrams, like
0.5 milligram per kilogram, which is about, I think I told her to take 50 milligrams a day. And it took her, I had her come back in a month. She was back to 2020
Wow.
I did a field test because usually when you have a trauma to the optic nerve, you get a field defect. And she did say that, like, when she was reading the letters, she could read all the letters, but there were some like little distortions to some of the letters. But she read them, and visual field test, pristine, no blind spots, no scotomas. And
have you ever had anything like this in the past? Like,
well, I never tried it before.
Had you had a recovery like that?
Oh no,
within the conventional realm.
I've never seen anything like that.
It's just, have you ever seen anything in the literature?
So that's what made me say it to her, was I had seen, like, I was researching when I was going to come out with the Methylene Blue product. I definitely had seen both red light therapy for nerve injuries and also for Methylene Blue for all kinds of neuropathies and things like that.
Ubiquitous.
So just, I don't do things based on like what was the p-value on that study. I think more like, well, what was the mechanism of action? How does this work? What, like, so it's so Hippocrates, who used to practice good medicine,
right,
the father of real medicine before we got
right
into modern times, he said he's the first person who went from just relying on myths and tradition, stuff like that, to say you observe, and you reason. Like, half of being a physician is actually putting the pieces together, to being the detective to figure things out. Mm-hmm. And so I think in things of mechanism. So I thought it would work because of understanding what red light does and what Methylene Blue does. So I just did it, and I knew that neither of those things was going to harm her. And I didn't have her, by the way, I did not have her look directly into the light. I had her just play on her cell phone for half an hour sitting underneath the light. So she's not directly looking into it, but she's getting plenty of red light to the eye.
How long did it take?
It was a month later I saw her back, and I don't know how long it took for her vision to grow up. I should have asked her.
You bring up a lot of really good points, and one of the things that I think frustrated me, as one of the heart guys in the hospital, one of the cardiac anesthesiologists in the hospital that I was working at during COVID, they put the 3 heart guys in the ICU, and you would hear a lot of, with certain patients, well, we're not doing that because there's no randomized controlled trial. For instance, N-acetylcysteine, right? N-acetylcysteine is a precursor to glutathione, and glutathione is what converts peroxide, as do we. It's what converts peroxide into water, and it's something that is highly useful at the level of the mitochondria. It's highly useful if you think about the mechanism of action for why COVID was so deleterious, is that peroxide was a big problem, especially at the pulmonary vasculature. So it made sense. And there is good data with NAC and the use SARS-CoV-1 and also influenza. Well, you would hear stuff. How many
people in good metabolic shape were in the ICU for?
It's a great point.
COVID.
None. I mean, none.
Not
right
many.
Now here's the problem though, is that you would hear, well, there's no randomized controlled trial, and my problem with that is that, to your point, is that in medicine not all of the knowledge that we have is predicated on a randomized controlled trial. You can gain knowledge through observation. I tell people all the time, I don't need a randomized.
We're relying on randomized controlled trials of people who have metabolic syndrome. That's where all of this stuff about lipid, the idea that you should be taking statin drugs, okay? Any evidence that it may be beneficial is only for people who are in deep metabolic syndrome, and basically not even that's even questionable. But the number needed to treat for statin drugs for people of low risk, if your only risk factor is high cholesterol, you have to treat 225 people in order to prevent one cardiac event, okay? And how many diabetics did you just make? It's like 3% of the people become diabetics from taking statins.
The numbers.
It's monetized sick care. It's not.
You bring up good points. You bring up good points. The numbers 0.2%. The reality is that in order to form plaque, there's 4 things, right? You need you need some type of ApoB particle to become oxidized. That sets off a reaction where it calls out for monocytes. So monocytes then attach to the endothelial cell of the vasculature. They move into what's called the vascular intimum, become macrophages. They engulf the macrophage, form a foam cell. Over decades, those foam cells form plaque. So you need ApoB particle, inflammation, oxidative damage, and vascular immune dysfunction. Where does that oxidative damage, inflammation, and vascular immune dysfunction come?
Diet.
Most of the time, insulin resistance.
If you don't eat an oxidized polyunsaturated fatty acid,
the caveat,
you won't have atherosclerosis.
The caveat to what you're saying is there are patients that have familial hypercholesterolemia where they're genetically predisposed to high ApoB burden, and they are metabolically healthy, and they still get disease. So there's a question, there's certainly a question, but my point is that
I don't think anyone's going to watch this because we're getting too nerdy.
Oh, please.
Yeah,
Part 2, 3.
But my frustration is that number one, there's this hyper focus on cholesterol only when it comes to vascular disease. When scientifically there's actually 4 different factors that are at play that are eventually going to cause plaque. And the other thing is that I even have patients in my practice right now, and they don't diabetics, folks with end stage disease, and they don't understand that this plaque that they have, whether they had a carotid, a surgery on their carotid or a cabbage or whatever, this plaque that they have is systemic, and it's been developing for decades. So the best time to treat somebody for plaque is when they're in their 20s, and the best way you treat it is through prevention, namely diet, exercise, sleep, stress. To get it back to optimizing mitochondrial function.
I heard a really interesting comment. It was I was watching it was Chris Masterjohn, the guy who has that MitoMe company. He was on Joe Rogan last week, and he was saying that he was, I don't know if it was his idea or he was just repeating it, but young people with high cholesterol, what's really going on is it is associated, it correlates with later on increased heart disease. But what his argument is that it's actually poor metabolic function, that those lipid molecules are not getting converted into the hormones that you need, the other things that are all made from cholesterol. And so, yes, you're going to see this elevated cholesterol and you're going to see the problems later. But like I was saying, the high cholesterol without the other risk factors, it's not lowering their cholesterol doesn't prevent the disease. Well, it's actually just a sign that they have poor metabolism in the first place.
To your point, with Framingham, which is where we got a lot of this data to begin with, the Framingham longitudinal study out of Massachusetts, I want to say the triglyceride to HDL ratio was 4-fold more associated with cardiovascular risk than was LDL, your quote, bad cholesterol. And the triglyceride to HDL ratio, as we know, is directly proportional to insulin resistance. In other words, in any patient, the higher that ratio becomes, the more insulin resistant the patient
is. Are these things which is the chicken and the egg? You're just seeing that as the marker for something, or is it a correlation? Or
No. So I think what you would deduce from that is that insulin resistance is a far greater risk factor for the development of plaque than is just simply an isolated LDL. Yeah. And that the marker for the insulin resistance in that study, because they're not measuring HOMA-IR, they're not measuring fasting insulin, they're not measuring C-peptide, they're not measuring leptin, they're not measuring any of these metabolic markers. They're looking at triglyceride to HDL ratio.
Why don't doctors measure oxidative LDL versus LDL?
Such a good. Well, we do. we look at LPPLA2. We look at oxidized LDL. We look at myeloperoxidase. We look at ADMA.
Because I think I'm guessing. I'm just a dumb eye carpenter. Eye carpenter.
Yeah, right.
Eye dentist. But I would guess that there probably is a correlation with ox LDL and cardiac risk.
Yeah, I mean, I personally, it's interesting. So
that's a whole nother podcast.
it's a whole nother podcast. Yeah, it's interesting.
Can I come back?
Oh, yeah.
I think we're gonna have to do one about the eye.
1,000,000%.
I don't think we did the eye.
All right.
Yeah, I just here's I guess my last question. So you measure. Tell me the name of the apparatus that you use to measure with the 3 different wavelengths for glaucoma. What was the name of that?
The ERG, electroretinogram.
The ERG. Okay. So if you have glaucoma and you have a reduced amplitude, then that's telling you've already got optic nerve damage.
Right, and it probably would correlate with visual fields or optic nerve thinning, like physical things that you could see.
Can you use that? Can you use that test to look at diabetic retinopathy or to look?
It has like a whole
macular
protocol
degeneration.
for looking for. It's mainly macular degeneration. So this circles back to where we started. They're all the same disease, but it's just presenting as different phenotypes. And so measuring electrophysiology of retinal cell function, it's going to pick up any chronic disease, any metabolic disease, and all diseases metabolic disease. So yes, it works. And what I see a lot of diabetic retinopathy, macular degeneration, glaucoma. Those are like the bread and butter of an ophthalmology practice. So yeah, I use it for all 3.
Interesting. And do you see in those 3 big diseases, is it more common in patients with chronic disease, patients who have been? It's interesting.
It's just their phenotypes.
Yeah. Yep.
It's the same thing as Parkinson's versus Alzheimer's versus depression versus all neuropsychiatric disease has its root cause in metabolic function. All human disease. It's a breakdown in turning food into energy, and then you see it. It's that radical oxygen species damage, which is also related to inefficient metabolism. I was going to go down a path that is like so fascinating, but I just want to metabolism. The solution is to get your mitochondria optimized. And it's not a pill. It's not like, it's primarily what you eat, when you eat. And fasting is a huge part of the when you eat. And then the aligning circadian rhythms is critical to, so what's happening in deep sleep, the reason why sleep is so important, that's when mitophagy happens. So mitophagy is just like autophagy, where when you go into a fasting state and other times, you're getting cell turnover. Right. Well, the same thing is happening in mitochondria. Okay, so mitochondria go through this mitophagy it's so amazing how much we know and how much it's all like readily available to us now with AI and stuff like that.
Yeah, all that.
The mitochondria, when they start to get more oxidized and they start to be more leaky of radical oxygen species, that means the enzymes are more and more oxidized, so there's more and more bottlenecks, and they're not transmitting the electrons down the chain properly. They put these 2 markers, PINK1 and Parkin, on the outside of the mitochondria, outer mitochondrial membrane. Then, while you're in deep sleep, is the main time, your cells go through and they recycle the old mitochondria and make new ones.
So just like autophagy, where they basically take all the parts.
Yes, it's not necrosis. It's not like the stuff dies. It's like you had a Lego that was put together, and you take apart all the parts, and now you put together a new one.
The functional parts.
Yeah,
it's
and
like they turn
now
it into a
you have a
junkyard.
fresh, young, non-oxidized mitochondria in there. So there's ways like my next. So this was working directly on the electron transport chain. So the next one I'm going to do is a mitophagy formula, that speeds up, and I know of some things that can signal increased rates of mitophagy.
What are those nutritional supplements?
It's top secret.
Oh,
That's in the next.
It's well known, but I'll save that for
Save
when we have the product.
This has been awesome. We carry InSurf. We use it. We carry it. We offer it to our patients. at Icaria, we are highly focused on mitochondrial health. All of our offerings with our longevity center deal with upregulating mitochondrial function, and our offerings with our Vibrant Lab panel are now really getting into the nuance as far as the three-pronged approach to the mitochondria. So, Val, I appreciate it.
He's got the most amazing red light bed I've ever seen. That thing, it looks like
Medical
a Ferrari.
grade.
You know, it's
cool.
pretty sexy for a red light machine.
Red light wellness.
That's
and
right.
It's a guy out of St. Louis who's a retired engineer and physicist. He would actually be cool to have on the podcast.
Yeah.
Super fun.
We'll just
guy.
have to do another one. We'll
For
do
sure.
one with the eye.
For sure.
We'll talk about the eye.
Next time we talk about
Well,
it's all the same disease.
we hit the mitochondria. That's for sure. So Val, I appreciate it.
Yeah, thanks, man. Thanks for having me on.
If you guys need a good ophthalmologist, we're developing a whole a group of practitioners that are really kind of into the metabolic health, into functional medicine, preventative medicine. So Dr. Zudans is one of those guys. We appreciate you.
Thank you.
Felice.
And that's it. Merry Christmas, Happy Hanukkah, Happy Holidays. We'll see you next time.